Hello, welcome to Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we are looking at the first ever core practical in the entire A-level biology course, and it also happens to be the first ever core practical in topic 1. So let's begin. Before we go ahead, the core practical in question is to investigate the effect of caffeine on heart rate in Daphnia. So let's begin. So starting off with the introduction, and this is basically to give you context of what this practical actually involves. So caffeine is a stimulant which increases heart rate by increasing the release of excitatory neurotransmitters, and you will look at neurotransmitters in greater detail in topic 8. But this is all you have to know for now. So this experiment will be using an organism called Daphnia, which is a very, very, very small organism, and you're very likely to see them in your labs in school. And they are suitable for this experiment as they have a translucent body, which means you can see through it, and you will be able to observe its heartbeat. So what is the equipment you need for this practical? So first off, you need Daphnia because that is the organism you will be doing the practical on. You need cavity slides, dropping pipettes, distilled water, caffeine solution of a range of concentration, cotton wool, test tubes, a stop clock, and an optical microscope. So now going on to the method. So dilute the caffeine solution with distilled water to produce several different concentrations, and place some cotton wool on a cavity slide. So the reason you're placing some cotton wool on a cavity slide is to reduce the movement of the Daphnia so that it doesn't escape. And add a single Daphnia to the slide to also make sure that it is alive. So how do you know if it's alive? Well, if it's making some form of slight movement in the water, then it is safe to assume that it is alive. But if it's really still and doesn't seem to move at all in the water, then it's very likely it might be dead. So do keep an eye out for that. And of course, when you look at the heartbeat, and if the heart isn't beating, that's an indication it's dead. So it's time to pick another Daphnia. So moving on, use the filter paper to absorb the water around the flea or Daphnia. It's the same thing, but and then you use a dropping pipette to add a few drops of distilled water to the slide. And do not use a cover slip. Why do we not use the cover slip? Because at the end of the day, the Daphnia also needs oxygen just like every single organism that exists. So you don't want to give rise to anaerobic conditions. So basically keeping the Daphnia alive by having a good supply of oxygen. So use a stop clock to time a minute and record the number of heartbeats. So there is one way we can do this. It's by using this thing called a paper trick. So what you can do is you can place a piece of paper on the table and basically make dots on this paper to the rhythm of the heartbeat of the Daphnia. So when you do that, you are more likely to pick up the heartbeats, even if the caffeine concentration is really high and the heartbeats are really fast. So you can still pick it up because after you make the dots on the paper, you can count up the dots, and that is the number of heartbeats in a minute. So now repeat the experiment with the various concentrations of caffeine solution, and you will see a trend in how the heartbeat changes with increase in caffeine concentration. So risk assessment. So there's 2 risk assessments to be worried about, to look out for, and one of them is the biohazard, which is the Daphnia itself. It is a living organism, so hence it would be a biohazard. And to minimize risk, use disinfectant when handling Daphnia and wash your hands with soap before and after the practical. And The second risk assessment point to be looking for is a broken glass, because you are using a lot of glassware in this practical. So handle all glassware in a safe and secure manner. Keep away from the edge of the desk, obviously, to minimize the risk of breaking glass. And of course, in case of an emergency, elevate the cuts, apply pressure, and do not remove the glass from the wound. And of course, seek medical assistance. So now for going back to the results. So when you collect your results from the practical you did, what you're gonna do is you're gonna plot it into a graph. So in this case, you'll be using a line graph. And in this graph, you will have the caffeine concentrations on the x-axis, because that is the independent variable, basically the variable that you are changing. And then on the y-axis, you will have the number of heartbeats, which is the dependent variable. So what's the dependent variable? Dependent variable is what changes depending on the independent variable. So conclusions we can make from this practical. So what you will notice is that as the concentration of caffeine increases, the heartbeat of the Daphnia will increase. And caffeine increases the heart rate of the Daphnia by increasing the stimulatory neurotransmitters. And as the concentration of caffeine increases, heart rate also increases. But this might not always be the case, because after the a certain point of passing the caffeine concentration, what you might notice is that the heartbeat begins to drop. So in this case, it is very likely that Daphnia is close to dying. So it's one thing I would suggest to improve this practical is after you finish immersing the Daphnia in a particular concentration of caffeine, return it back to its environment and then start again. So that way the Daphnia is not overstimulated and it's not put under pressure, pain, because once again, this comes under the ethical side of using Daphnia in this practical. And of course, and finally, after the practical, ensure the Daphnia is returned to its natural habitat. So that is all from Professor Snap today. So hope this video has been useful. This is the first ever practical that we've covered together in this channel. So if it's a good format, if you've enjoyed this video, if you found this video useful, please do let me know in the comments below. Or if you think there's some other points I could be covering in these practical videos, then do let me know in the comments as well. And don't forget to like, share, and subscribe. And thank you so much for watching.